So I thought I would talk a little bit more about the homework today, spend a little bit of time giving some tips on how to implement the different techniques we're learning about. So we've seen several different classification techniques. K nearest neighbors is a really simple method. Often does not quite as well as some others. We've seen cross-validation. We're using the bootstrap, the lasso, so several different things, and wanted to speak to a couple of tips on how to actually do it with our data. So when you download the data, homework 6 data.csv, I think I already have it. Oh, I already have a script. Let me open this. Nope. There it is. Okay. So when you download the data, remember that we have genes, kind of pretend genes, features, we could say, in the rows, and samples in the columns. And the class memberships for these training data are given by the column names, or just by the description in the homework. So A indicates class A, and so on. You have 225 training observations. That's the first 225 columns. The last 75 are for testing. You hold them out to evaluate the model later. Okay, so a couple of issues that I've seen on the discussion board. The bootstrapping, let me just comment on this. There was a comment on the discussion board about doing the bootstrap. The idea here is we're going to use K nearest neighbors. We're going to pick the value of K that seems to be an optimal one. And then using that optimal K, apply KNN to it. So get the KNN fitted model for that optimal K. With that optimal K, when you look at the confusion matrix, that tells me the misclassification rate, and it can also tell us more specifically how did we do by class. Were we more accurate for some of the classes than for others, for example? But those are just estimates. That's just what I saw as an estimate. So just like an average or a mean of a bunch of numbers is an estimate, and so it's variable, it has its own standard deviation, it'd be nice to have like a confidence interval for a prediction error estimate. We estimate 92%, plus or minus 1.5%. Would be even better than just being able to say 92%. That's what you're doing in part 4, is trying to use the bootstrap, which is a general tool for getting a standard error estimate. And then you can have a confidence interval on the interval. Okay, so this code right here is meant to be what you would put inside a loop while you're doing the bootstrap. So over in R, I'd say capital B is whatever we're going to do. I don't remember. We'll say 1,000, 100. Okay. Say 100. So for B in 1 to capital B, create a bootstrap sample. I don't know why I'm writing this on the whiteboard. Just write this in a script. So let's say B is 100. We're going to do a bootstrap that's like for B in 1 to capital B, create a bootstrap sample that we're going to work with. And creating the bootstrap sample is what you can use this for. What this is doing is randomly picking with replacement from all the training A samples, and randomly picking with replacement from all the B samples and the C samples. So when I put all of those together, all those indices, all those columns, I have a new training data set. It's a bootstrapped version. It's a resampled version. And then I did the same thing for the test data. So at the end of that, we have Y train. So let's say create a bootstrap sample to get Y train and Y test, like as described in the assignment. So inside the loop, once I've done this, these sample commands here are randomly picking. That's the idea of the bootstrap. You randomly sample with replacement from your data. So this needs to happen every time inside the loop. I randomly sample to get a new bootstrap sample. And then from there, you just proceed as if that's your data. And you apply whatever method you wanted to apply. Apply method and store the statistic that you're interested in. Do you want a standard deviation for the test error, for example? Then you would fit the model on the training data, which is your new Y underscore train, apply it to the Y underscore test data, and get a number. So like test accuracy, the Bth1, let's set up a placeholder up here. Test accuracy is repeat nothing B times. Then the Bth1 would be the proportion of the Y test that we just created, the new bootstrap version, that were correctly classified when we built a model with Y train that we just created. Apply method to the two objects that you just created. After you do that, that's a generic kind of, that's almost a generic representation of the bootstrap. As in, you could do this with regression, you could do this with t-tests if you didn't trust that you had a normal distribution. You wanted to get a better standard error, a non-parametric standard error. You could bootstrap your data, compute a t-statistic, compute that 100 times, and then if we compute like the square root of the variance of that, of all of those, that's an estimate of the standard error of the statistic. Whether we want to do inference on a median or a regression coefficient or anything, you can formulate a bootstrap and do a non-parametric analysis. The bootstrap's really cool, and it's very generic. You're using it here specifically just to get an estimate of the statistic that is test error, or test accuracy, sorry. Estimate test accuracy for each of your 100 bootstraps, and if I were to draw a picture of those 100 bootstrap accuracies, here's a histogram of those 100 bootstrap accuracies. So maybe the one we saw was right there, probably 92%. But there's random variability in any estimate. So the standard deviation I could estimate from this. What's the standard deviation of these 100 values? For you statistically interested, more inclined folks, this is a non-parametric picture of a sampling distribution. So, and sometimes you can theoretically say the sampling distribution is, say, normal or something. Bootstrap's nice because you can do it without having to make assumptions or without having to have huge sample sizes. That's my tip on doing the bootstrap. How's it coming? Just give me the confidence interval. So if you get once you get the standard error, standard deviation, do whatever you saw. Well, yeah, you could either use the original accuracy, or what I said here was to average all the accuracies that you saw, which is fine. It's probably a better estimate. So that plus or minus basically 2 times the standard deviation is a typical 95% confidence interval. It's just we want a better standard, or we don't even have a standard deviation unless we do the bootstrap here. This is like a way we can get one. You sniffled. Is it any of these I'm happy to make, say, extra credit, I don't want them to kill you in terms of R programming. I think they are good exercises for understanding.
Bootstrap on my computer runs in like less than a minute. It's done.
Okay.
But number B takes 50 hours.
50 hours, okay.
I'm on Windows and I can't do batch. It's not easy to get batch to work.
Okay.
So that's, and I'm running an i7 with 32 gigs of RAM.
Okay. Well, then how about for D? If you've got code that's thoughtful, as in you thought it through and as well as you can tell it does what we want it to do, but you don't have time for it to run, submit it and I'll tell Yabu to use, if you did the problem, fine. I don't want the computation time to be a limiting factor. So part D, just to formalize that, minimal requirement is a well thought out and reasoned bit of code that you think would accomplish what we need. If it's obviously wrong, if you put no effort into it, we'll be fair. But if you tried hard and it looks reasonable, that's fine in terms of credit as far as I'm concerned. Encourage you to not give up trying, because this won't be the only time if you ever use R again. It won't be the only time you need to do something that's computationally intensive. So things like possibly dumping computations to C. There's a learning curve on learning how to do that, but a lot of really complicated or really memory-intensive stuff can be done really quickly and with pretty simple code in C. Running in a batch mode is nice, especially if you have access to a server, like in your research department or something. If there's a server that you can log on to and submit jobs. Or on campus, there's high-performance computers that we all can easily get accounts on. And I've got an R program that'll run 2 weeks or something. I can submit that as a job, like with an R batch type thing.
Do you know how to make batch work on Windows by any chance? Because it's much more complicated than the way you talked about it in Q&A. It doesn't work.
Doesn't work. I haven't done it on Windows for years, but when I did do it, I remember having difficulty because of my memory was like the path variable, where when I went to the command line and I wanted to say r.command batch ... it didn't know r.command, and I didn't know how to make it. Yeah. There's somewhere that you have to, I think, modify a path-like variable, which means whenever you type a command in the prompt, it knows what that means, because it's got a list of all the functions on the computer that actually run stuff. So somewhere there's a path variable, like in a modifications setting, and then you put like a colon and then type in the working directory of R.
Yeah, you have to like code the path to the execution.
Mm-hmm.
R, if you go and
That's one way to do it. If you add it to the path, then every time you just type R, it knows what you mean. That's one of those things that you do once and you forget how to do it because you don't have to do it until you get a new computer. Yeah. Linux and Mac, it's easy to set up the path thing, and then, yeah, it's easier. Another, I guess, pro tip is an option for working with really big data. R can have a hard time reading in enormous data sets, as we've seen, I think, and it bogs down with repetitive calculations. One option is to use another software package, as a secondary tool that is better with big data sets, like SAS or JUMP. JUMP is, in my experience, much smoother at loading big data sets and doing simple stuff with them. Same thing with SAS. There's a JUMP license on campus. Every student can get it, I think, for free. It's super easy to use. It's point and click. What other questions on the homework? What can I help with. There were questions about naive Bayes on the discussion board. Let me talk about that for a second. So you have examples in the conscript, and let me just emphasize a couple of things. When we read in these data, let's see. Bless you. When we read in the data, we set aside training and testing, and we created a couple of data frames. And the data frame format is a column for each individual. So the first, column, a row for each individual. And so in the first column is the class membership variable. And in the remaining columns are the features or the genes. It's a little confusing. Which way should you organize your data? And I mentioned early on, if when we interact with methods that come from classical multivariate statistics motivations, the paradigm is to put individuals in the rows and variables in the columns. Naive Bayes operates like that. So it's expecting individuals in the rows, variables in the columns. That's why we're putting it this way. So I noticed on the discussion board there was some, when these were being used, like when naive Bayes was being used, in particular for predicting, I think, there were like dimensions don't match or something. The length of this is not the same as that. I think that has to do with make sure that the data you pass in are in the right format. So the way I set up the con data was to have a data frame, which these types of functions like, because now I can put like a column that's a categorical variable that has class membership, and then separate columns for all my variables, which in this case are genes with individuals in the rows. How's that? Is this off the charts? Hard or something? I don't know. Okay. All right. Are there any questions? All right.
I do have one question. So when we were doing when you were talking in the Q&A about doing the TT loop, basically to make all those T calculations that you have to do every single time.
Yeah.
That's what's bothering the whole thing.
Yeah.
So.
And that's a technical thing, but the slides specifically emphasize that point. You have to include that in this cross-validation, or you get biased estimates.
But when we coded it, we just took the like 175 and 76 to 150. But shouldn't it really be like where GRP equals A compared to where GRP equals B? Because you're taking one out of one of those groups, so you really just want to put a curve.
Yeah.
And so if you do 75, and you're going to take 74 of A and then one of B.
Yes.
Training set.
Yeah. So that would, to get it implemented correctly, that's a detail that has to be. Yeah. Easy way to do it would be, instead of trying to keep track of which one is, how many are in each class, to do like you said and just say, compare where group of whatever's in my current training data equals A to where group equals B. All right. I wanted to talk some today about another technique that is commonly used. So we've learned about like linear discriminant analysis as a classification technique. Quadratic discriminant analysis is same thing, except you allow for a little more flexibility. Naive Bayes is linear discriminant analysis, where you assume all the features are independent. Instead of a covariance matrix, capital sigma, that you have to estimate covariances between all umpteen thousand genes, Naive Bayes makes the very, very simplistic assumption that they're all independent. Almost certainly bringing in lots of bias, but remember that overall goodness of a predictive model has to do with both bias and variability. So the general principle is that with high dimensional data, omics type data, where P is much bigger than N, you often have much better overall performance in like mean squared error type of interpretation by taking much simpler models at the expense of bias, but in the goodness of getting lots less variability than, say, a neural network, which is a really complicated, highly tuned model that, while it's super accurate, it's also going to be very highly variable if you were to redo your experiment with 20,000 genes and 30 rats or something. So lots of variability. Anyway, LDA, QDA, naive Bayes, three flavors of the same idea. They're all trying to mimic that Bayes rule, the Bayes classifier, best we can do in terms of minimizing error rates under a normal distribution assumption. We also saw the lasso as a feature selection technique. If we're willing to postulate a model for, say, the gene expression, mean gene expression or something, as a linear model, I won't write that stuff out today, but as a linear model with a separate covariate for all of my thousands of genes, then there's thousands of coefficients, those betas. Lasso shrinks those betas to zero, some of them, in terms of which of you are really helping to discriminate. Let's minimize the influence of the others. And it actually shrinks them away, many of them. It is a real feature selection process. So you go from thousands of features that you want to predict cancer with down to much fewer, maybe 100 features that are selected for because they help. I mean, they effectively discriminate. Okay, another common technique that's pretty different from what we've seen, but has some nice properties. In particular, it can be very intuitive. And this is the idea of classification trees. What am I doing here? Don't want this. These are not like linear regression models at all. These are not like regression models at all. So these create tree-like representations for a decision. If you want to, for example, estimate the salary of a baseball player, tell me how many hits they had, tell me how many years they've played. Let's predict salary. This would be what a tree-based prediction model would look like. It doesn't look like what we've seen. What it does is says, first off, is the years variable less than 4.5? If it is, then I'm going to predict 5.11. If they had at least 4.5 years, then ask that question, did they have less than 117.5 hits? If so, my prediction is 6. If they had more than that, my prediction is 6.74. So it's that kind of thing is really easy to explain what the classifier is doing. So it's highly interpretable. And you can also gauge from this picture the relative importance of each of those breakpoints. The most important distinction in terms of what made the most difference in classifying was the first one because of that bigger distance. So we don't have to know all the details about this, but that's the essence of what a tree-based classifier does. So that's what's nice about a tree-based classifier, the kind of simplicity, and I'll show you the flexibility of it to be able to capture kind of complex, nonlinear boundaries. What we'll see, though, is that in practice, that simple interpretable version of a tree method often is not as accurate as most other methods. There are ways to remedy that, called boosting and bagging. If you ever heard those phrases in the machine learning context, it's ways of averaging models. Like, let's do lots of tree-based models by bootstrapping from the data and doing a separate tree each time, say. And then my final prediction is going to be the average of all those predictions. That's a way to kind of resample. Boosting / bagging are techniques for doing that with trees. And you end up getting pretty accurate methods, in my experience, but you lost the simplicity of the model. There's no longer one tree that you could look at and say, this is what my classifier is doing. Okay, so simple, useful for interpretation, not always the best. Bagging and boosting. Random forests is a tree that has been bagged or boosted, I don't remember. It's one of those applied to trees, random forests. So random forests are often very accurate in complex settings, but they're not easy to interpret. Whereas like linear discriminant analysis is easy to interpret. Naive Bayes is really easy to interpret. Like, what's the effect of gene 12 on my classifier? It's a beta coefficient for gene 12. So I could like say these are the ones that seem to be driving the classifier. Remember that with statistical learning, we could have either a numerical variable that we want to predict, like blood pressure, or we could have a categorical variable, like cancer or not. The authors here call those two regression and classification, respectively. Here comes regression. So this was that example that I initially pointed to. Let's try to predict baseball salary in terms of two variables. We might say, let's try to predict blood pressure in terms of two biomarkers, maybe. The algorithm or an algorithm results in this as the optimal choices to make in terms of minimizing mean squared error. Okay. So hopefully the tree idea is easy to kind of see what it's doing. Which of these is true? If it's one thing, go left. If it's the other thing, go right. And you end up with a prediction. This, when we remember in that homework 7 that I brought up briefly, I talked about drawing those decision boundaries, and we looked at LDA as not curvy enough to capture the curvature of the boundary that we created. LDA, QDA, they're all drawing like circles and ellipses to define boundaries, or lines in the LDA case. So lines or ellipses. A tree can produce boundaries or reproduce boundaries that are not like that at all. This is not a line, this is not a curve. It's two line segments that break the space into 3 quadrants or regions. That's what's unique about trees, is that they can attack problems that don't that you can't do well with, say, a linear model or some of the more linear-based things. let me just show you a couple more slides here. Okay, there's an issue of overfitting. If you have lots of variables, like we do in an omics setting, thousands of features, then a tree will often result in overfitting in the sense that it'll use all of your variables. It'll tend to use all your variables, just like we'll tend to overfit a model if we only look at resubstitution accuracies, if we apply it back to the training data. That's why we like cross-validation and test error. Same thing with trees. They will tend to overfit, and so the next few slides are about what they call pruning. So moving it back some to get rid of needless detail. Okay, and you do better when you do that. You have to tune the model, just like we've had to tune like the lasso, we've had to tune everything we've done. You specify a model, and then you have to do something to pick which flavor of it you want. Cross-validation is a generic technique that you can use to do that. So you can use it in a tree context. And let me just show you one slide about this random forest idea, and then we'll go see what how to use the random forest. Okay, so trees are nice, but they often are not accurate. They don't compete in terms of accuracy. But the idea is that if you were to somehow scramble your data and refit the tree and let it kind of fit the intricacies of that scrambled version of your data and do it again and do it again and again and again, you'd get lots of trees. And so for every prediction you need to make, you run it through all of those trees and get the prediction of all of them and average them. That's the idea of aggregating trees, and that's all that this boosting and bagging is about, or that's what it means. I'm not going to go into further detail on it, except to say that random forest is, okay, actually it's not bagging, it's an extra trick, but it is the same flavor. So it's a resampling based approach. You get lots of trees, you make lots of predictions, you put them together into one kind of master prediction each time. Hard to interpret the results for that reason. So let me show you what it looks like in R. Here are the con data. I think we've talked about the lasso code. Okay. There's a variety of things you can do with trees. A simple tree like you could reproduce was in the slides. No boosting, no bagging, no funny stuff. Can be from this function tree in the tree library. There is a cross-validation function that searches for, it tunes the model. Every one of these things we need to tune it somehow. How much shrinkage with lasso, what's the threshold for LDA, in this case, how much, quote, pruning should we do of the model. And so this thing will tell us what a good choice is. Just like you picked the value of k for the k nearest neighbors classifier using cross-validation. How much, quote, pruning should we do? And then random forest comes from its own package, and it operates a lot like the naive Bayes function does, as in it expects kind of a regression formulation. Response, tilde, covariates. We have everything set up in a data frame where the first column is the class membership variable, the group variable. Here it's called GRP. And the next so many are for the different features or genes. This thing, I think you can leave it as a default. If there's not a default value for mtry, go ahead and use 2. That's okay. Or read the book for a better choice. They talk about what this means. It has to do with random forests. They do a model aggregation thing. So they do lots of trees using the bagging idea, resampling, fitting tree to each time. The problem is that there's correlation in the predictions among all of those kind of bootstrapped trees because the same observations are in them a lot of the time. What random forest does is tries to decorrelate those predictions and then put them together. mtry has to do with how many variables to allow. Okay, I'll point you to the book. Okay, pick mtry equal to 2. That'll be fine for the purposes of the homework. If you say importance equal true, that gives you back something you can use to score or like rank the features in terms of how important are they to the model. You'd like to have a accurate classifier. You'd also like to be able to interpret the classifier, as in, here's 100 SNPs that if you genotype them, I can predict cancer. Well, tell me what those SNPs are so that we learn something about what biologically is there. Here's a classifier based on 2,000 gene expression numbers that predicts whether you're going to have a stroke or something. Out of 30,000 that we looked at, the important ones were these 1,000. So you need, whichever method we're doing, we'd like to be able to get some kind of score like that. Random forest has its own score, and I'd have to point you to the book to know exactly what it is. But it gives us a number that speaks to the relative importance of each variable in the model. Are there questions about implementing the random forest code? Is this assignment too much? It depends on background totally, because this class has a wide variety of people in it from different backgrounds. So I'm happy again to I don't want this to be a killer of R programming. So if it is, let me know. Let's see. Any other questions about R code for the homework? All right. Well, what I intend to do next, unless you guys have more questions about this prediction stuff, are there any special topic requests for prediction? Because I could spend another lecture on something if there was interest. Otherwise, what I was going to do was move to protein mass spectrometry. So there was some interest on the discussion boards for how do you analyze mass spec data. So you get mass spec data is interesting because you're trying to quantify proteins instead of genes or microRNA or anything. And you can't put intact proteins on, say, a microarray very, very well. I think there are microarrays, but I don't think they have the kind of performance that people would like. So a method of choice is mass spectrometry, but what you get is not data on proteins. You get data on peptides. Kind of like in next-gen sequencing, you get reads that come from genes, and you have to kind of work your way back to the gene level. Mass spec has that by going from peptide to protein. All right. Are there any questions further? Otherwise, I think that's what I wanted to cover today. Okay. Have a good weekend.